ladies and gentlemen, our focus today is going to be on DNA mutations. So hopefully you know by now that DNA inside of your cells codes for the production of proteins. Well, sometimes the DNA can get altered, changed, or mutated for one reason or another. Now, what we're going to do today is explore those mutations and understand how they can have an impact on the protein that is produced inside of the cell. So hopefully by the end of this video you should be able to differentiate the different types of mutations. Hopefully you'll also be able to predict the impacts of mutations on the production of a protein inside of a cell. Just a few vocab terms for this particular video. We're going to talk about mutation, specifically mutagens, point mutations, frameshift mutations, and silent mutations. Let's get started. So mutations themselves are simply changes in an organism's DNA. And if you think about it, we've talked about the importance of DNA in the production of proteins. So when you alter the person's DNA, you end up changing the protein. It has an impact on transcription, and as a result has an impact on translation. Now, a mutagen is something that causes a mutation, and there's a variety of different mutagens that are out there. UV rays, smoke, radiation, just to name a few external mutagens. Now, what's interesting is that the DNA inside of your cells is actually being mutated quite frequently. But fortunately, you have biological mechanisms inside of your cell that prevent them from ever being expressed as proteins in your body. The cell undergoes a process called apoptosis, where it actually destroys itself if it thinks or knows that the DNA is actually altered inside of the cell. So mutations can be quite dangerous, but your body's pretty good at keeping them in check. Take a look at some of the mutations over here. We've got a deletion of a letter, so normally it would be A, B, C, D, E, F. Well, B just happens to get deleted. A duplication, an accidental duplication of part of a DNA. Sometimes you can get inversion and translocation as well. So there's a variety of different mutations that are possible as a result of external as well as internal factors. So the two major types of mutation that we are going to talk about today are point mutation and frameshift mutation. Now, a point mutation just simply exchanges one nucleotide for another. So if you take a look at the diagram on the right, we have CTG, AGT. Well, instead, a point mutation occurs with the A, and so we end up getting CTG, CGT. Okay? So that's an example of a point mutation. We exchange one nucleotide for another. Another one is known as a frameshift mutation. Now, this is an insertion or a deletion of a nucleotide. So rather than exchanging one for another, we insert one in. So for example, in between the A and the G there, we've inserted a T. Or one happens to get deleted. And so in the bottom example, the A gets deleted. So those are frameshift mutations, and they're called this because it actually shifts the frame of the codon that is being read by the protein when this becomes RNA. You'll see what we mean here in just a moment. Some examples of point mutations. If we take a look here, our normal RNA sequence, AUG, CGC, GAA, UAG, has been changed to CCC. This means that the amino acid should be arginine, but instead is proline. So one example of a point mutation is we change one amino acid in the sequence. So instead of an arginine, we have a proline. Now that is very problematic for the cell. This can also create a premature stop codon or change a stop to another codon, which could have even more significant impact on the cell. If we have stop before we should have stop, then the rest of the amino acid will never be produced. And also if we change a stop codon to a regular codon, then the sequence is going to continue. The amino acids are going to continue to be added to the protein. So premature stop and altering a stop can have a significant impact on the amino acid sequence. We can also have what's called a silent mutation. So for example here, CCC codes for proline, and the last C gets altered and changed to an A. Well, that also codes for proline. So we change the sequence, but we don't actually change the amino acids, because if we look at our chart, we'll recognize that CCC and CCA actually code for the same amino acid. So this is known as a silent mutation because it changes the sequence but doesn't change the amino acid. Now, this one is probably the most dangerous, and we'll talk about this. This is a frameshift mutation. So this is your insertion or your deletion. So for an example here, we actually inserted an A in between the last two C's, where it's coded for arginine. So instead of CCC, it's CCA, and then now that last C becomes the start of the next codon, so we shift the frame of the codon sequence. Now the reason this is more problematic is because this changes every amino acid after the mutation. So it's far more dangerous because with a point mutation, typically we just change one amino acid in the sequence, but with a frame shift, we're changing everything after that insertion or deletion. And it can be far more dangerous because it alters more of the DNA sequence, which alters more of the RNA sequence, which ends up altering more of the amino acids that are produced in the protein. Now, keep in mind that mutations in sperm or egg cells can be passed on to offspring. Those are inherited. But mutations in body cells do not pass to offspring. So for example, if I happen to get a mutation in one of the cells inside of my body now, the only way it can be passed on to a child or offspring is that if it's in sperm or egg cells. If it's in body cells, that cannot be passed on to offspring. So hopefully you understand a little bit more about mutations and how important it is to be able to understand that they are changes in DNA that are caused by mutagens. Keep in mind that point mutations exchange one nucleotide for another. They may cause a change in one amino acid. They could be silent. They could cause a premature stop codon as well. Also understand that frameshift mutations insert or delete a nucleotide, therefore changing every amino acid after the mutation. Hopefully you've got a really good grasp of this. We'll talk more about mutations, how they happen, how they occur inside of the body, and what the results are of those mutations when we try to produce a protein from that amino acid sequence. All right, guys, we'll talk to you later. Bye-bye.